So welcome back friends, welcome in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to talk about three analytical questions from recombinant DNA technology. So this will going to be a video series and it is the first video of that video series. So today we will going to talk about three important analytical question from recombinant DNA technology. Then I will try to bring more questions like that. Okay, so let's start. So first questions for today is the restriction enzyme EcoR1 cut the DNA at the sequence GTT-AAC, and restriction enzyme HAE3, which is a quite unusual one, cuts DNA at sequence GGCC. So they told us to predict how frequently will each enzyme cut the double-stranded DNA. So here they only wants from us that we will derive that how after what base pairs or after how many base pairs we will get our recognition site. That is the type 1 number mathematical problem that I have told. So here as nothing is mentioned, so you should choose GC content as 50% and AT content as 50%. That means the amount of AT-GC is equal in the entire genome. So as EcoR1 recognizes a 6 nucleotide recognition site, so the every 4096 base pair after there will be a recognition site for EcoR1. Okay, similarly as HAE3 is a 4 base pair cutter, so they will recognize one recognition site after 256 base pair. I have told you that when there is nothing mentioned that GC content is 30% or 80% content is anything, that means when there is not mentioned that GC content is either less than or greater than 50%, okay, then you can use this formula where we are considering that GC content is 50% and we are simply using the formula 4 to the power n, where n is the number of nucleotide present on the recognition site. Okay, so that will tell you the base pair after which you will get your gene of interest, okay, or you will get your restriction site. Okay, so by deriving the two answers, it is very easy to understand that HAE3 will appear more frequently than EcoR1, right? So now let's move to the next question, that is quite interesting. That so I would like to recommend you to first read the question very carefully, then go for the solution. Okay, so you are given a bacterial plasmid DNA. Okay, we are having a bacterial plasmid DNA carrying tetracycline resistant gene. Okay, no problem. Wait a minute. So you are given a bacterial plasmid. So this is our bacterial plasmid, and in this bacterial plasmid there are a tetracycline resistant gene. Okay, so this is my tetracycline resistant gene and a ampicillin resistant gene. Okay, very good. Okay, now they are telling you that you have a IKB DNA fragment with blunt ends. That means they are telling about your gene of interest. Okay, so this is your gene of interest, and they are telling you a restriction enzyme E. So there is a restriction enzyme E who cleaves the plasmid vector DNA within ampicillin resistance. That means there will be a cut site for restriction enzyme E within the ampicillin resistance, right? producing a single linear DNA fragment. And I have already told when I have taken the class of restriction mapping that if there is a one cut site present in the circular plasmid and you treat them with that particular enzyme, you will end up with a linear double stranded DNA, right? Because this cutting will simply linearizes the circularized plasmid. Clear? Now, how do you insert the given DNA fragment? That means they are telling you to insert the given DNA fragment into the vector at e-recognized restriction site. Okay, that means the question is we have to insert this gene of interest into that E site, okay, to produce a recombinant plasmid. Okay, and how do you select the transform E. coli? So this is very important question, because this will clear the concept of your recombinant DNA technology in various aspects. Okay, so let's try to solve this one. So for solving this type of question, I would first like to recommend you to go and pause the video first and try to answer it on your own, because I have already uploaded videos on the different concept that is necessary to solve this question. So I believe that you can. Then please look the solution, then it should be helpful to you. Okay, so let's start. So they are telling us that we have a plasmid. This plasmid have two restriction site. Okay, very good. One for ampicillin resistance, another for tetracycline resistance. And they are also telling us that we have a gene of interest. So we have to cut this gene of so we have to insert this gene of interest inside this vector. And they have also given us another important information that there is a restriction site for enzyme E. Okay, and this is a blunt end cutter. Okay, so the first important concept that this question fulfills for you, that is, if you cut this plasmid with a E restriction enzyme, then you also have to cut the plasmid with E restriction enzyme. Okay, and remember it is not possible that a restriction enzyme will generate both blunt and staggered in. Okay, that is another concept that here fulfills. That means E here not only cut and generate a blunt in, but also E also here generate a cut which will be a blunt ended cut. Okay, clear? So we have to treat both our gene of interest containing source and our plasmid vector with the same restriction endonuclease, that is E. Okay, and as this is a only one cut, because they have told us that only one restriction site present in the plasmid, so our plasmid will be linearized. Okay, so now they want to told us that you have to join these two. Okay, so now you can here use linkers, adapters, or anything, right? You can use linkers. So what is linkers? Linkers are nothing but a short stretch of nucleotide which have restriction endonuclease cut site for different enzyme, different restriction endonuclease. Okay, so suppose if you use a EcoR1 cut site, so EcoR1 linker, so there will be a short stretch of nucleotide which containing GAA TTC CTTAAG. So this nucleotide block will be attached here, this nucleotide block will be attached here. Similarly in the case of your gene of interest, right? So now there is a problem. Why there is a problem? Because suppose in the case we don't know what is the sequence of gene of interest. Okay, and by chancely there are also a GAATTC CTTAAG inside the your gene of interest. So what do you think? That your restriction endonuclease will only cut your linker, or it will cut also these things also? So obviously the second possibility will be higher, that it will also cut the gene of interest, right? And as a result, your gene of interest will be lost, right? So we will not recommend the using of linker. Similarly, adapter using is also not a good choice, because adapters are nothing but ready-madely cutted linker. Okay, so ready-madely cut product of the linker. That means they will give you ready-made staggered in. But there are also a problem, because when you will add adapter, your plasmid vector and your gene of interest together in presence of DNA ligase, so in presence of DNA ligase, the self ligation of adapters are more thermodynamically favorable than the ligation of gene of interest and plasmid with the help of adapters. Okay, and as a result, your entire work will be lost, because of those adapters will join together and convert them back into your linkers. Okay, so that is the thing. That is why we will not going to use linker or adapter. Instead, those things we will going to use a different thing. That is, we will use the terminal deoxynucleotide transferase. So I think you have watched my enzyme in recombinant DNA technology video, and if it is, so I think you know that terminal deoxynucleotide transferase is an enzyme that can add nucleotide template independently from the 3' end. Okay, this is a special type of polymerase that have the ability to add nucleotides template independently at the 3' end. Okay, so now if you provide the building blocks, that is DTP here, I have chosen DTP. That does not mean that DTP is only applicable. You can use DATP, you can use DGTP, you can use DCTP. It is totally on your choice. But remember it, if you use here DGTP, then you should choose DCTP here. Okay, here I have chosen DATP, so as a complementary here I have chosen DTP. Okay, so that is the idea. So by which you can create a staggered end. Okay, now by using T4 DNA ligase, you can easily join them, isn't it? Right? So now our ligation, that means our first question is solved, right? Again, I am repeating what I have done here, that they are telling us that there is a restriction endonuclease present on the ampicillin resistant gene. And as this is a only cut site present for the restriction endonuclease E, so upon digestion by restriction endonuclease E, we will end up with a linear DNA. Okay, so now after that we will treat them with terminal deoxynucleotide transferase in presence of DATP. That will generate a 3' end or ready-made 3' end for you. Okay, and here also, here I have forgotten to write about the E, so sorry for that. So there will be the first step of restriction endonuclease digestion by E, then terminal deoxynucleotide transferase treatment. Okay, that will give you this thing, and now just by using ligase you can join them together. Okay, so we have successfully completed the first question. Now they are telling us that how you can screen them. So now I want to tell you that as gene of interest is inserted here, that means ampicillin resistance are no longer ampicillin resistant, they become converted into ampicillin sensitive. And this is not only applicable for any kind of antibiotic resistant gene, but it is also applicable for any kind of genes that are available. Okay, suppose if we talk about that there is a restriction endonuclease cut site present on LAGZ gene, so upon insertion of the gene of interest, the activity of the LAGZ will be inactivated, right? So that is the idea. Okay, so that means now our recombinant DNA will be ampicillin sensitive tetracycline resistance. Okay, on the other hand, our gene of interest was ampicillin on the other hand, our plasmid vector was ampicillin resistance tetracycline resistant. Okay, now we will simply culture them in presence of LB agar In case of cloning technique, LV agar is usually preferred. So you have to write LV agar in presence of tetracycline resistance. So why I have not written about ampicillin resistant here? Because remember it, there can be a chance of self ligation of the vector in presence of the DNA ligase, okay, instead of the insertion of the gene of interest. Okay, that means in the conical where our bacterial cell and vectors are present, there can be both recombinant plasmid containing host present, along with there can be also normal plasmid containing host present, because after the ending of this procedure we will do transformation step, right? This transformation step is nothing but insertion of the plasmid inside the E. coli via a process called heat shock calcium chloride. Okay, and this is the number one preferable method for transformation of the E. coli, right? So now this plasmid is inserted inside the E. coli, right? So E. coli will get two different type of things. One is your plasmid containing gene of interest, another one is only your plasmid. Okay, I hope you are getting this, right? Because of the chance of the self ligation. Okay, so now you have to screen them. So I should say that the bacteria who have acquired only normal plasmid, they have ampicillin resistance, tetracycline resistance property. Okay, on the other hand, the bacteria who have acquired recombinant DNA containing plasmid, they should have ampicillin sensitive tetracycline resistance property. Okay, now you are culturing the total broth culture you have in the Petri plate. So as here both type of things are present, ampicillin sensitive tetracycline resistance and ampicillin resistance and tetracycline resistance, so we will plate them in presence of only tetracycline resistance, so that all kind of colonies can grow, both this one and the upper one. Okay, now via a process I think you know called replica plating, we will create a replica plate. So what is the process replica plating? So replica plating is nothing but a process where we are taking the replica of the colonies present in the mother plate. So the first plate that we have prepared are known as mother plate. Okay, so how we will take the replicas? So we will take the replicas with the help of a special type of instrument known as velveteen cloth. Okay, so as a result, in the velveteen cloth there will be replicas of the colonies. Okay, now we will transfer those replica colonies on another plate containing LB agar along with tetracycline resistance and ampicillin resistance. Okay, and we will incubate them. Okay, so this kind of plate that is prepared by taking the replica from the mother plate are known as replica plate. Okay, and that is why the name replica plating is given to this technique. Okay, so now upon incubation we will get colonies, but they will be less in number than we get in the mother plate. Okay, and this is obvious, right? Because in the mother plate both ampicillin resistance, tetracycline resistance plasmid containing bacteria is present, along with ampicillin resistance, tetracycline resistance containing bacteria are present. Both kind of bacteria are present in that plate, but in that plate there are only the presence of ampicillin and tetracycline resistance. Okay, so here I have also made another thing. So because here you have to only add tetracycline, not tetracycline resistance. Tetracycline resistance is the gene present on the plasmid, right? And that is why bacteria are getting the ability to survive in presence of the antibiotic. So you have to add tetracycline antibiotic. Similarly here you have to add both tetracycline and ampicillin antibiotic. Okay, so the colonies that we have get here are nothing but the colonies for the normal plasmid containing cells, right? So now we can easily say this, that in the replica plating all the bacterial colonies that are present are having only normal plasmid, right? And all the rest of the colonies that are present on the mother plate are having ampicillin resistant, ampicillin sensitive and tetracycline resistance colonies. Why I am telling this? Because remember, we have taken the replica of the mother plate and we have used those replicas to construct the replica plate. That means the colonies, the position of the colonies here will exactly be the same with the mother plate. Okay, so the position of the mother plate where colonies are present, but in that same position in the replica plate the colonies are missing, we should say those colonies are nothing but your recombinant DNA containing bacterial colonies. Okay, that means now by comparing replica plate and mother plate we can easily screen those bacteria who have my gene of interest. Okay, so I hope you have understand this very well, right? So again repeat this question, again I am repeating this question. So in the question they are telling us that you have a plasmid containing ampicillin resistance and tetracycline resistant gene. Okay, and now there is a restriction site present for restriction enzyme E. Okay, and they are also telling you about a gene of interest of length IKB, which is your gene of interest. Okay, and your job is to insert somehow this gene of interest inside that vector. Okay, so now the problem is they have told you that this restriction endonuclease E is a blunt-end cutter. Okay, that means they will generate blunt-end for you. So as according to the theory of molecular cloning, if you are digesting your vector with E, that means you also have to digest your gene of interest with the same restriction endonuclease. Okay, so you have to digest the gene of interest with E. They will generate blunt-end cut site for you. Okay, so now by taking those things they are telling you to how you can insert those gene of interest with the vector. So now I think you will tell me that what is the problem, just add ligase and then it is over. But it is not that, because the ligase which we are mainly using are E. coli DNA ligase. Okay, which usually prefer your sticky end ligation. Okay, blunt end ligation can be done with the help of T4 DNA ligase, but it is difficult than the sticky end. Okay, so under such circumstances we will try to make it easier. So I have told you about the linkers and adapters, but I have also told you that if you are using linkers and you don't know what is your gene of interest sequence, then that could be dangerous, right? Because there can be a chance of the loss of the gene of interest, right? Similarly, you cannot use adapter because ligations or self ligation of adapters is thermodynamically favorable. Okay, so we also don't use adapter. Under such circumstances we will take the help of terminal deoxynucleotide transferase in presence of DATPs. If you are using DATP here, then you have to use DTTP here. Okay, the complementary one. Then by using the ligase you can simply ligate it. Okay, and you will get a ampicillin sensitive tetracycline resistant plasmid. Now you will transfer them in E. coli, right? Now inside the ligation mixture there can be a chance of self ligation. That means instead of inserting the gene of interest, plasmid can self ligate. Okay, under such circumstances you will get plasmid having ampicillin resistant gene, tetracycline resistant gene. Okay, so the E. coli will get both the type of the plasmid. Okay, so now we have to screen them. So how we will screen them? We will simply plate them in a plate consisting of LB agar along with tetracycline antibiotic. Again I am repeating this, right? It is not tetracycline resistance term, it will be tetracycline antibiotic. Okay, so under such circumstances, if we will incubate them, as I have told you that both the recombinant DNA and your normal plasmid are tetracycline resistance, so both will grow in the plate, there is no doubt about this. Okay, so now after that we will take the help of replica plating. What is replica plating? Here we are using velveteen cloth to take the replica of the colonies from the mother plate, and we will transfer those replica colonies onto another plate, and via incubating we will get some colonies. In the second plate we have used LB agar along with both tetracycline and ampicillin antibiotic. That simply means that the first type of plasmid containing bacteria will grow, but second type of plasmid containing bacteria will not. Okay, but as we have taken the colonies from mother plate and with the help of replica plating, that means the position of the colonies in the replica plate and the mother plate will be exactly same. Then by simple comparing of mother plate and replica plate, we can find out the colonies who have recombinant DNA containing bacterial cells. Okay, so that is how you can screen them, and this kind of screening is known as secondary screening. Okay, so the tetracycline who are selecting both the plasmid containing bacteria, both kind of plasmid containing bacteria will be known as primary screening, and the ampicillin will be known as secondary screening. Okay, because it ultimately separates for you your recombinant DNA containing plasmid. Okay, so that is the today's video are getting too longer, so I have to stop this here. In the next video we will going to discuss about this question. This is also interesting one, so we will talk about this later. Okay, so thank you for listening to this class.